So part 2 of our introduction to protozoans. So remember, they are the first animals, considered the first animals, and these are the protozoans, which are the animal-like protists. Within the kingdom Protista, which there are other versions, the plant-like and the fungal-like. So there's probably about 50,000 animal-like protists. So yeah, and that took place 1 billion years ago, not a million years ago. So we're going to deal with using energy. And we'll look at how they actually move as an example of how they use energy. So the amoeba uses that pseudopodia. So if you draw an amoeba here, so those pseudopods are these finger-like extensions here. So there's a couple kind of mechanisms it uses to get the pseudopod finger to actually move. So remember, there is the endoplasm towards the inside of all of these. And then you've got the ectoplasm, which is kind of on the outside. So it's differentiating the types of plasma, cytoplasm that it has. And so there is something called a gel-sol theory. So the gel-sol theory talks about the solubility of those two areas. So gelatin is going to have less water, and the soluble one will have more water. So it's a way of changing the percentage of water in the two areas to get it to be more soluble and more, it flows more to get that cytoplasm to move into that pseudopod. And the other thing that it uses is actually these proteins. So we blow this up a bit. It's two proteins in particular. That's an actin protein, and you don't need to write this stuff down if you don't want to. And then a myosin protein, which is a walking protein. Kind of walks around. And so these microtubules are the cytoskeleton that are inside here. And so these myosins will walk and kind of push those out to extend that. And we have that in all of our cells. The myosin kind of carries stuff around inside of our cells, all sorts of organelles. So that's how it can extend a pseudopod, or it'll break the bonds in between to get that pseudopod to absorb. All right, so we're talking about that. But let's watch the video here. So what you see in the video is that, and this is a sped up. So you see the pseudopod extending, and it's squirting the cytoplasm in there. So this is happening more real time. So it's a little bit slower of a movement. So you've got the proteins acting here on the edge, and then also your ectoplasm and your endoplasm. I don't know the specific mechanics of that gel sol theory. And then we get to cilia, the quick beating. So these guys, the paramecium, kind of got a little bit more defined shape. So what they have are all of these little hair-like structures. So that's what your cilia is. So they're hair-like. And so they can move either way, back and forth. It's in the membrane they've got these hair-like follicles that extend back and forth, and they move kind of in unison, moving back and forth to create that flow. And these guys move pretty quick. So compared to the amoeba, which is kind of slow and lumbering, these guys are moving around fairly quickly. And when you get to that small of a size and you're in water, it's like trying to move through pudding. And so they can actually get a lot of traction and a lot of movement.
Paramecia swim happily in ponds and streams throughout the world. Their quick movements are produced by an army of hair-like oars that beat to the rhythm of intracellular chemistry. Called cilia, the tiny parts with purpose create water currents that bring a constant supply of gifts in the form of bacteria and other organic matter. Entry into paramecium begins in the oral groove, which leads to a bustling chamber called a food vacuole. An unsuspecting bacterium might frolic for a while in this churning chamber, but its days are numbered. Correction, its minutes are numbered.
And then we get to the flagella, those whip-like. So in the euglena here, it's going to have this long tail, and that's what the flagella is. And plural would be flagellum, and the verb is flagellate. And it's a pretty interesting, I mean, when you look at the whole complex, it's got some basal cells, and then basically like a rotor that's going to spin around. And so figuring out this mechanism has taken a bit, types of cells, how it actually generates momentum. And a lot of that has to do with creating an electron differential. That looks terrible. Mine keeps, told it I was left-handed. So differentials are ways to create basically engines. And your chloroplast does this, mitochondria does that. We use it to generate electricity. It's a lot like setting up a hydroelectric dam. We get a barrier, we put a lot of particles on one side. They want to go to the other and create an equilibrium. What we do is we channel that movement. So the water will come through these tubes in the hydroelectric dam, and as it flows down, it will spin a turbine. Once you get kinetic motion, once you get movement, we can turn that into electrical energy. So we take a magnet, some wires, copper wires. So moving magnetic field will create an electron flow. That's all we do. Same thing here. We create an electron differential between the two membranes, and as it flows, it causes it to spin. And if it wants to go, so it's going to spin around in a circle, and I believe that clockwise will create
forward motion, and then counterclockwise, it can go in reverse as well. And so we see that with these Dublina here, when they're kind of whipping that tail right there, moving that situation. But it can, sperm use this, a lot of bacteria will use these flagellum, just spinning. It's kind of like the motor of a boat to create that movement forward or backwards. It is faster than the pseudopods. Between cilia, I don't know which one's faster. So then how they obtain energy. So we're going to be looking at either heterotrophs or autotrophs. So the amoeba is going to gather its energy and eat through this process called phagocytosis. Which is a type of endocytosis. You remember from Bio 1 when you talked about that, osmosis or passive transport, fusion, and then active transport. So endocytosis is an active transport. You're bringing in a big amount of stuff. Literally means endo-cell action. Endo inside, cyto cell. Penocytosis is when you gulp in a bunch of water. So from there, it's going to go and digestion will occur. So once it's engulfed with its pseudopods, it makes a food vacuole, and that's where we're going to have digestion. A lysosome will probably fuse with that, inject a bunch of enzymes that will break down whatever tissue they can get from that, back to the building blocks, use it to make more cellular processes, and then anything else that is not needed, the waste materials, they will excrete through excretion or exocytosis. And that can happen pretty much anywhere in the amoeba, any one of the arms. So there's no definite mouth or anal opening to do that. So here's an amoeba eating a paramecium. So the paramecium is moving around pretty quick, but with those slow arms, it can kind of engulf, close that off, and then it will, the paramecium is going to go crazy with all its hairs trying to get out of there. And then this is going to start to fold in now once it recognizes it's got a food particle in there, and turn this into a food vacuole that they'll bring that lysosome in and start to digest that, break it down. And poor little paramecium, smothered alive. Now the paramecium here, they have kind of that, they use an opening designated for channeling this type of food. So it goes to that oral groove. That's where it starts. From there, it'll move to the mouth. Then on to the gullet, which we'll see that in a lot of our organisms as we see how they eat and stuff and their body parts. They'll have gullets, kind of where they're, if you don't have teeth, how do you break stuff up? And then that will produce a food vacuole. We have digestion occur, and then from there, it'll go, the waste materials will go to the anal pore and be excreted that way. So what's really interesting here about the video is you're going to see that beating motion feeding food into the oral groove, the mouth, the gullet, and then into a food vacuum. So here what we see is a combination here, and so this food vacuole is getting a bunch of whatever it can fit, E cells, something like that. So the food vacuole will get full on different size, and it'll pop that off, and then it'll go to be digested through lysosomes. There we go. And then we'll go ahead and start building up the other. It's just kind of like a constant process. And then you'll get to the anal pore, which is just south of that oral groove, and kick that out. And I'm going to flip back. It'll let me. There we go. So these two are heterotrophs. They are the other feeders. So hetero means other, homo means the same. So hetero is different. So its troph, nourishment, comes from other sources. Strictly. They're the strictly animal-like type of protist. There's fungal-like, and then there's plant-like, which we'll get with the euglenas. So the euglena here, this is going to go through, there's a self-feeder. Which is what we call an autotroph. So automatically, auto means self, an automatically driving car, automatic shifting transmission. It shifts on its own. It's not a manual. You don't need to do that. So they are getting their nourishment through that process of photosynthesis. Which is, come on. So photo means light or a photon of light. Syn means together. And then the action. So using light to put things together, and this is kind of the brilliance of life, I think, on this planet. The first life forms, early 4 billion years ago, where did they get the energy to do work, to maintain themselves, to do homeostasis? Well, they got it from other chemicals. So, first life were autotrophs. And they used chemicals. And they found chemicals that had high energy in its bonds. And that energy came from the heat of the earth to generate those hydrogen sulfide compounds. But that limited life to only existing where there were heat sources. We still see that life today. If you go to Yellowstone National Park, go to hot springs, they'll have chemoautotrophs at the bottom of the ocean and near these ocean vents. So finding a way to find another energy source, photosynthesis is amazing. The fact that you can take light this photon of light, trap it, and use it to basically take a carbon and break it apart. The CO2, break that apart and add that to water. And that's where you get your carbohydrate. You hydrate that carbon, and then those carbon-carbon bonds that are now created, that's got a lot of energy. It's a high energy bond.
And again, it uses those those electron potentials. It's able to use light energy, and it's got certain molecules and proteins that can harness and grab those and transfer that to exciting electrons and creating a differential between two membranes. And then that movement allows for you to do that. And it's going to stay there in that bond
until that bond gets broken. And that's the brilliance. When you break that bond and you release that energy, that is respiration. That is
and the cool thing is it's a paired. You take this arrow, flip it over the other way, and when you consume sugars, take in oxygen, and you break it apart, your byproduct is carbon dioxide and water, which is what plants need. So it's that paired relationship. Those chemoautotrophs, they couldn't do that. The arrow went one way. And the byproducts were some other molecules. And so it didn't just easily reverse. So here you've got a reversible system that's constantly bringing in energy and then releasing energy. Bringing it in and releasing it.
You didn't see the. So the euglenas, they can experience algal.
High spot, you can go find that light source to speed up the photosynthesis, which was definitely an improvement over this.
There where the light is. Homeostasis. So homeostasis is the act of keeping your internal environment consistent. So a lot of these guys are living in an aquatic environment, and a lot of times it's freshwater systems. So freshwater systems have a lot of water. Versus saltwater systems, you have a high solute concentration, which means you have less water. So if you remember from osmosis, it wants to go from high water to low water concentrations. So, and this applies for all three of them. So if I'm a single-celled thing, and I'm in an environment with an abundance of water, I mean it's got a high level of water. And there's less water inside. What's going to happen to try and water's going to try and equate itself, so it's going to keep trying to force its way into that membrane, into that cell. And it's small enough to get through the membrane, just squeeze through. If you damage this membrane as a single-celled creature and it falls apart, you're dead, you're done. Or if this just keeps swelling up with water and bigger and bigger and bigger, it's going to eventually pop open, lice. It's going to break open. So these contractile vacuoles, they work to maintain the water pressure. So you've got the contractile vacuole there. U-O-L-E. And then around it you have these star-like, I think they're called spongiomes. And so these guys kind of take on the water, push it into here, and then the contractile vacuole will then take that excess water and squirt it out through a pore in the membrane. So it's like if I'm going to keep taking on water, just like a boat,
funnel it to the bottom of the boat where the bilge is, and the bilge is going to be a water pump that's just constantly pumping it out. And that's kind of what these do. So it can happen very quickly. Where squeeze and see the spongiomes kind of fill up immediately and put that water back in. Just you see the spongiomes, see that projectile vacuole filling up, squeezes that water out. The water that's picked up in the spongiomes squeezes back in. So it's that constant kind of water pump. Keep taking on water, funnel it, get it out. Funnel it, get it out. Otherwise, the whole thing could, without a cell wall, plants don't need it. They got a harder cell wall that kind of keep the shape of that cell. And they want that water pressure, actually, that osmotic pressure. Usually, big ones have two. So your euglenas, your parameciums, they're going to have two contractile vacuoles. Amoebas can have more than, because they get really big. An amoeba will also, instead of just squirting it out through a pore, they'll actually, pinocytosis, they'll just use a membrane. Fuse that vacuole with a membrane and then release it that way, exocytosis. But that's the way that they can maintain that water pressure. And that last bit is evolving. So, as we notice, these are our zoos, protozoans. So we consider these proto means early. Zo means animal. It's our prefix for animal. And an means like. Like, having a quality. So it's an early animal-like thing. And that's what these are. So the euglena is considered to be a plant. It has the mitochondria. So how did this happen? Well, it's through that process called the endosymbiotic theory. Endo is inside, sim is together, bio is life. So it's life working together, one went inside the other. And so this happened a while ago. So the endosymbiotic theory there, I thought I'd go in front. So what happened was this aerobic bacteria, so it was really good at taking sugars and digesting them and creating energy, gets taken in, invaginated, taken in, absorbed, and instead of decomposing it or digesting it, they work out that symbiosis, where that bacteria now turned into mitochondria, and it's been doing that so much that it doesn't have to worry about all the life processes anymore. It reproduces, it uses energy, but it's now in a protected environment. It doesn't have to dealing with the outside world, finding food. All it's doing is producing energy, an abundance of it that the larger cell can then use. The larger cell is the one that's dealing with the outside world, bringing in food, getting rid of waste, but it doesn't have to worry about getting any energy. Converting it. That's being done by this mitochondria. So all eukaryotic cells have that mitochondria. That came first. And later, photosynthetic bacteria were absorbed by other cells. So that's where we get our plants and animals. The animals did not do that. The plants did.
in that photosynthesis. So it's really good at capturing light, turning it into sugars. And that's where you get kind of that picture there. So the mighty mitochondria, it is a double membraned. It's got a membrane, and then it's got this other set of membranes here. And it takes in sugar, breaks it apart. It's going to release carbon dioxide and water, but it's also going to create ATP, adenosine triphosphate. It's got 3 phosphates. It started with ADP, diphosphate, 2 phosphates. So it took the energy from breaking apart that sugar and charged this up to here. Added that phosphate back on. ATP is just going to go, it's like the universal battery in the cell. If I need to add membrane, build stuff, break stuff down, move things. ATP is what you used to break that phosphate off, the energy released, you use it to do something. Then it goes and gets recharged. You can even go down to AMP, monophosphate. You can break off 2 of them. They just get charged back up. So the photosynthesis, the chloroplast, same thing. Membrane, and it's got a bunch of these thylakoids, there's stacks of them. They look like a bunch of quarters stacked on each other. And what they can do is they can take in and capture light, the CO2 and H2O, and rearrange those using the energy from the light and give off those sugars. So the cool thing is that these were their own living things billions of years ago. And the hint that they were is that they are both what we call double membraned. No other organelle inside of a eukaryotic cell has 2 membranes. ER doesn't, Golgi body doesn't, nothing else. Vacuoles don't, plastids, just these 2. So this exterior membrane is back when it was its own living thing, and then it has an internal membrane, like organelles. Same thing. This was its exterior membrane back when it was its own thing, and then membranes for organelles to do the job. So double membrane, they are self-replicating. So mitochondria make more mitochondria, chloroplasts make more chloroplasts inside of a cell. And what do you need to replicate? Well, you need to know how to replicate yourself, and that's also the cool thing. They both still have their own DNA. So there's DNA within mitochondria that is how to operate and be a mitochondria and make more mitochondria, not in the nucleus. Same thing with the chloroplasts. It's their own DNA from billions of years ago with how to operate. So the DNA inside of your mitochondria has been there for billions of years, passed down every time it replicates. Now, where does all my mitochondria come from? Where did I come from? How did I get made? Sex. My parents, right? So my traits, my eyes, hair color, skin color, or lack thereof, all came from the genetic material from my parents. Where did I get my mitochondria from? Both of them? One of them? Only one parent gave me my mitochondria. All my mitochondria came from one of them. Which one?
what makes sense? Mom. So when we look at sex cell formation in humans, what we have is, my mom is going to give me the egg. And then my dad, this little itty bitty cell. So half of this comes from my mom, and the other half comes from my dad, inside the nucleus. So when this fuses here, and then it gets taken straight to the nucleus. All this other junk, come on. All of this junk came from my mom. So that means every mitochondria in here came from my mom. So I can trace it back. It came from my mom, it came from her mom, which came from her mom, which came from her mom, and her mom, and her mom. What we've actually done is we've traced back mitochondria. There's changes every now and then, mutations, so there's some mitochondrial differences among people or other organisms. And we have traced it back to a mitochondrial Eve that came out of Africa 35,000 years ago. So if you're not from Africa, you're from some other part of the world, your heritage is. Your mitochondria, we can trace it back to 35,000 years ago. One of those, about 6 women, I think, that left Africa and gave rise to the majority of the rest of the people on the planet. The greatest genetic diversity in our species is in Africa. There's more genetic diversity between 2 Africans from different tribes than there is between 2 other people anywhere else on the planet. You take me and some random Chinese dude, you compare our DNA, we will be closer genetically than some tribe from the north and southern part of Africa. They will have more genetic differences between them. Cool tidbit, we've used this to solve crimes, determine lineages. There's a situation, this really nasty dictator in a Spanish-speaking country, Argentina, or might be Spain. Yes, Spain, yeah. So, as dictators are wont to do, they don't like people that are rising up against them. So he was a dictator. They were enjoying pretty liberal views until he took over and used the military might. So a lot of college-aged and younger adults were protesting, distributing pamphlets, stuff like that. And so the military would come in and basically kidnap you in the middle of the night. And they'd take you somewhere, put a hood over your head, take you somewhere, torture you, and kill you. Now, if you had kids, they wouldn't necessarily kill the kids. So they would have these kids and not know what to do with them. So they would just give them to people. A general's girlfriend. Hey, here's a kid now. Hey, you got a kid that's 7 years old. Here's another one. Just raise that one as well. Did not take any records. They just took people and disappeared. Called los desaparecidos. After a little bit, he was overthrown and killed. Then we had this issue of where do these kids belong? Their parents are dead. There's no records of where they came from. And it was the grandmothers and the grandparents that were like, we want to find our grandchildren. So the grandmothers got together, they realized, well, we look at their mitochondria, which came straight from their mother, which then went to their mother. So these grandmas all made, supplied their DNA, their cells, and they did mitochondrial analysis on the DNA. And so they could take a kid, look at their mitochondria, and match it with their grandmother. And I think they matched everybody back together. It took a while with genetic technology at the time, but it worked. There's also a chromosomal Y atom. So that is it. Any other questions?